We will get started. It's top of the hour. Welcome everyone to the seminar series of the Trans-NIH Resilience Working Group established in 2019 and led by the Office of Dietary Supplements as a coordinating activity to help harmonize language and conceptualize frameworks and resilience that can be applied across the NIH. The theme of this year's seminar series is experimental designs and outcomes that advance the study of resilience in the biomedical sciences. Today, we are pleased to welcome our speaker, Dr. Heather Whitson, Professor of Medicine, Director, Duke Aging Center, and co-director of the Duke UNC Alzheimer's Disease Research Center. In these roles, Dr. Whitson is an active contributor and thought leader in the emerging field of physical resilience as she strives to optimize independence and resilience in people with multiple chronic conditions. Today's presentation is entitled The Science of Bouncing Back from Health Stressors, Duke Pepper Model of Physical Resilience. With that, I will give Dr. Whitson the floor.
All right, thank you so much, and thank you for that introduction and the opportunity to speak to this group. I think I want to start by saying that, as noted, I'm a geriatrician and a clinician.
In medicine, and I am so excited to talk to this audience, partly because I think it involves a lot of disciplines different than mine, and that for me is always an opportunity to learn. And to have this construct that I'm going to tell you about that we've developed for a very clinical medicine purpose, really informed by our colleagues from a lot of different disciplines. So I think that the concept of resilience seems to resonate across every discipline that I've ever encountered. And I think that there are so many opportunities to use that concept that resonates to sort of inform or elaborate how it can be used and applied in our own work. So with that, I also want to make it really clear that I am speaking today sort of about a construct that has been elaborated by a lot of people, not just myself. I am at Duke, and the theme of our Duke Pepper Center and our Aging Center is resilience and age-related changes in resilience. And so what I'll be talking to you about is sort of how we've thought about this concept and how we've applied it, but very much recognizing that other people have applied and used even the terminology slightly differently. And I like to think of that as not a hindrance, but just an opportunity for discussion. Much of our work is funded by the NIA. I'm also an employee of the Durham VA.
Disclosures: National Institute on Aging, P30AG028716-11, UH3AG056925, P30-AG064201-02, R01-AG062623-01A1, U13-AG054139, R33-AG057806, P30-AG072958-01. Veterans Administration, Durham, Virginia. Geriatric Research, Education, and Clinical Centers, or GRECC.
And just since in this virtual world, it's I think it's kind of good to just place ourselves in the real world. And this is Durham, my home, and that is the Durham Bulls ballpark stadium, which is kind of the center of our town. And Duke campus is over there, not too far away from downtown Durham. That's Duke campus, which has this beautiful Gothic buildings, and you can see our beautiful chapel there in the back. And then in the inset, you see the chapel as well as the entrance from the campus side to the Duke School of Medicine. You can walk through these doors and be in the School of Medicine. And sort of behind that beautiful Gothic facade, there's this very modern looking medical campus, and I'm sitting right there. And in the Duke Aging Center, we're home to a lot of NIA-funded centers that conduct work on resilience and on other aspects of our theme. So the Duke Pepper Center and the PRIME Collaborative, or the Physical Resilience Indicators and Mechanisms in the Elderly, are particularly related to resilience. The theme of the Duke UNC Alzheimer's Disease Center is changes across the lifespan that contribute to the development or progression or experience of Alzheimer's disease, and resilience certainly comes into play in the work that we do there. And then we also house the Reach Equity Center for advancing healthcare equity, and we think about resilience also in that lens as well.
Duke University's Center for the Study of Aging and Human Development is home to NIA-funded centers such as the Claude D. Pepper Older Americans Independent Center, also known as the Duke Pepper Center.
So for today, I was going to tell you just sort of the Duke Pepper Center framework that we've elaborated, and then two approaches that we've developed to quantifying the degree of resilience in this framework. We focus a lot on provocative tests or stress tests as predictors of real-world recovery to real-life stressors. And then I'll give two examples of clinical studies that we're conducting that apply the framework.
The two clinical studies are PRIME-EEG and PRIME-KNEE.
So again, because I'm a clinician, the framework that we use for resilience is very much based in sort of clinical reality and clinical utility. And so I'm going to start with a patient case. And in this case, the patient is my father. So that is my dad there. He's now 77 years old. In this picture, it was just pre-COVID. So he was about 74, 75. And those are my two sons. And my dad relocated after retirement along with my mom, and they both live nearby. And we have kidded him that he had changed it. He turned in his title of CEO for the title of MVG, or Most Valuable Grandparent. And that's because he's a robust man in his 70s and spends a lot of time with my two boys. So not just the things that they enjoy doing together are fishing and horseback riding, but he also shuttles them around a lot to various sporting events and performances and things like that. So the clinical story that I'm going to tell you is that one time, just right before COVID happened, all three of them, because they spend so much time together, all got the same GI virus. And we've all sort of known the frustration of a GI virus, that you feel terrible for about 12 hours and then the symptoms disappear. And that's exactly what they all went through. But about 36 to 48 hours after the symptoms had disappeared, the boys were right back at it. And I went to meet my dad, who had taken the boys to a baseball game where one of them was playing in the baseball game. And I went up and sat next to my dad in the stands after work, and he looked sort of peaked. And I said to him, how are you feeling? You look like you're looking a little pale. And he said, yeah, he said, I'm over the GI virus, but he said, watching Chris out there run around those bases like nothing ever happened, and knowing that we were both sick as dogs just a day ago, he said, it just makes me realize I don't bounce back like I used to. And it's really that sort of that clinical reality, which is that what we see is people experience some kind of a stressor. And it could be, in this case, a GI virus. It could be something else like a surgery or a hospitalization or a heart attack. And oftentimes they will recover, but it seems that people recover in a different pattern. So some people, it takes a little bit longer. Some people may not recover as fully. And some people don't get knocked down as far, and then also have a slower climb back. So it comes from this very clinical place of thinking about how different recovery from the same stressor can be for different people, and in particular for us as an aging center, how predictably that changes with age. We saw this obviously play out in the COVID pandemic, both the fact that the hardest hit population were those who were over age 80, And yet, even within that age group, we saw incredible heterogeneity. And this is a person quoted saying, we had a 96-year-old guy who never even had a symptom, recovered like nothing ever happened.
Joshua Yu, a Penn Medicine geriatrician who is medical director of a West Philadelphia nursing home that had the city's first coronavirus outbreak, said about a third of the 22 residents there with confirmed coronavirus were asymptomatic, a third had mild symptoms, and the remainder got very sick. 5 died.
So what is it? What is it that drives, and for us, we're mostly interested in what is it that drives at a physical, molecular, or cellular level? What is it that drives that difference in recovery when a body is stressed? So considering 2 patients that are being evaluated for, say, an elective surgery, like a total knee replacement, we might think about 2 different patients that are coming in and are essentially the same chronological age. So a 76-year-old woman who's a caregiver for her husband. She has obesity, hypertension, depression. She leads a pretty sedentary lifestyle. She's had a history of coronary artery disease and is preparing to undergo this surgery. And the same day, you might evaluate a person who's 75. This person has well-controlled hypertension and glaucoma, and that's their only diagnoses. They play golf and tennis and have a supportive wife and 2 daughters. So we could imagine if we were going to try to predict.
Photographs of a 76-year-old woman and a 75-year-old man, and a graph depicting time on the x-axis and function on the y-axis. A vertical line at the beginning of the graph indicates the occurrence of surgery. A horizontal yellow dotted line represents A. A red line with a U-shaped trajectory represents B, and a gray dotted line with a downward trajectory represents C.
Which of these 2 patients is going to follow which trajectory, B or C, after the surgery? Most of us would probably predict that the gentleman is going to follow a trajectory like B. Almost everybody who has an elective knee replacement obviously has a period of time when they're less mobile, unable to do as much for themselves as they recover. But we would expect that he would recover back to baseline, if not even better than his baseline when he had the knee problem. And then every so often we see a patient who undergoes an elective surgery and just never gets better. And from that point on, the family will always refer to mom was never the same after that knee replacement, or mom was never the same after the stroke or the hip fracture. And so much depends in aging on how we bounce back and if we bounce back. And most of the time, we could get it right if we took 2 patients like this that had such different sort of baseline reserve going into the stressor. But sometimes we don't. Sometimes we would get it wrong. And in fact, the woman would sail right through the surgery. And this gentleman, who appeared to be a great surgical candidate, has complication after complication and never quite recovers. So again,
Duke Pepper Center Conceptual Model of Physical Resilience. This slide depicts a yellow oval, which represents the environment. Overlaying the oval are 4 boxes representing a linear model. From left to right, the model begins with the PreStress Reserve, including cognitive, psychological, and physical reserves. Between the pre-stress reserve and resilience phase are stressors, which include acute illness, injury, surgery, and psychosocial stressors. The subsequent phase, resilience or dynamic response, includes the range of response. After resilience is outcomes, which includes survival, independence, quality of life, and morbidity. The environment, pre-stress reserve, stressors, and resilience all have opportunities for intervention.
Our whole model is kind of based around trying to understand what are the things that we could measure that could help us start to tease apart to predict what it is that causes one person to have a good recovery or a not so good recovery. And then ultimately, where can we intervene? What are the opportunities to be able to intervene and produce better outcomes for more patients? And we really break it down like this. We think about what the person's reserve is before the stressor. We think of this as a very modifiable condition, not a fixed trait, but a modifiable variable of reserve. And we think about reserve could happen in different domains. Examples of domains shown here could be their cognitive domain, psychological domain, physical domain. And we certainly realize that even putting these into different domains is arbitrary because they're all linked, right? We fully believe as geriatricians that people's cognitive health, psychological health, physical health, other aspects of health and well-being are all very much linked. But importantly, we think of this sort of pre-stress reserve as something on a spectrum that changes. Then the stressor happens, and certainly the magnitude of the stressor determines how much the person's response is going to be good or bad. And then we focus on this sort of dynamic response, a thing that happens at the time of a stressor, and that we watch a dynamic process play out of resilience. And ultimately what we think is that when that dynamic process goes well, favorably, meaning the downturn is minimal, the upturn is complete and rapid, that we would think that that should then predict better long-term outcomes like survival, living independently, quality of life. This is the model. And all of this happens within the context and environment in which the stressor happens and the person lives. So the reason that we focus so much on this as kind of a dynamic thing of resilience after the stressor is that we also really think of resilience as this sort of ability of a biological system to maintain an equilibrium or to regain an equilibrium after it has been perturbed from its equilibrium by some sort of a stressor. And we think of human health as this kind of constant balancing act where we are seeking an equilibrium or a homeostasis, and that there are constant mechanisms in place to find that equilibrium despite these constant external stressors and perturbations, challenges that knock us away from an equilibrium. So we think of every person as this sort of complex dynamic system that is a integrated and overlapping set of subsystems that are sort of constantly in motion, moving, transitioning, adapting to changes in environment, and that essentially our ability to live depends on our ability to respond robustly and briskly to those changing environments and stressors. And we can think of the person's subsystems as their various organ systems, but underneath that, there are these cellular network systems, molecular systems, all the way down to genes and the activation and deactivation of genes. And then that person lives within another complex dynamic system, which is their social networks. So this is all sort of part of this framework of how we think about resilience. As I said, we recognize that with age, it is fairly predictable that our ability to respond after a stressor declines, but the rate of decline is not the same for everyone. And we're very interested in this question of why. I always show these photos because these are two gentlemen that are the same age. They're both 75. But again, you can certainly see that despite the same chronological amount of time on this earth, they appear very different in terms of what we would call their pre-stressor reserve. So that you could imagine that if they experienced the same stressor, the gentleman on the left would likely do better, because we think of a person's resilience after the stressor being constrained by, not 100% determined by, but constrained by what the reserve is before the stressor.
The word resilience is depicted in a central bubble with the following terms connected in a ring around the central bubble: cancer, metabolic disorders, neurologic disease, autoimmune diseases, infectious diseases, and vascular diseases.
And we think of this sort of vicious cycle where if we think of a person's resilience as that capacity, what's going to happen after they're stressed, we know that the accumulation of diseases over time diminishes their resilience to be able to withstand the next disease or the next stressor. And then lower resilience makes them more vulnerable to that next disease or stressor. So we see this as kind of a vicious cycle between the accumulation of disease and comorbidity, chipping away at resilience, and then making a person at higher risk for more morbidity. And then another thing that we think a lot about is the Geroscience Hypothesis. And I wonder, this may be something that has been discussed previously in this group, but really the idea of the Geroscience Hypothesis is that Aging itself, the aging phenomenon, is something that is mediated by these changes at a molecular and a cellular level, and that there are a few biological processes, and I've listed them here.
The word resilience is depicted in a central bubble with the following terms connected in a ring around the central bubble: response to damage and toxins, mobilizing energy stores, regulating inflammation and immune response, cellular damage control, protein folding and recycling, stem cells and regeneration, and genetics and DNA repair. Pillars of Aging adapted from Kennedy et al., Cell 159, 2014.
That these biological processes change over time, and that more favorable biology in these what have been termed the pillars of aging essentially determine the biological age of a person, which may be very different from their chronological age. And we believe that it determines or underlies a lot of their resilient capacity as well. So in sum, kind of what do we see as the influencers of recovery trajectories after a health stressor? I think that we fully acknowledge that, as I said, the magnitude of the stressor, no matter how robust a person is, there are some stressors that they would succumb to. We know that a person's attitude and mindset have a lot to do with how they respond to a health stressor. Their health status before the stressor, so that issue of their sort of reserve before the stressor, the support systems that are around them, the care and interventions that we as a healthcare team provide, we hope have something to do with how their recovery trajectory goes. And then that biological response, or what's at sort of the molecular cellular level that is allowing them to respond.
Regarding stressors, the question is, do you bend or break?
So one of the things that we think about a lot is whether this biological mechanism that underlie recovery after a health stressor, whether it's possible that there are different biological mechanisms that are most important to how vulnerable a person is before they're stressed.
The same graphic taken from the Duke Pepper Center Conceptual Model of Physical Resilience, as shown in slide 11, depicting PreStress Reserve, stressors, resilience, and outcomes within the environment.
And perhaps different biological mechanisms that play a bigger role in the response to the stressor. So for this, we sometimes use this metaphor of a castle under siege. So I'll tell you the way we think about it is that if the body is a castle,
Cartoon of a medieval castle under attack by knights.
We might look at this castle before it's attacked, and we have some inclination that the castle may not hold up very well to attack. So you can see that there is some frailty in its structure. Structurally speaking, its walls look a little old and damaged. Some of it has been replaced from rock to wood, and we think that might not hold up as well against an attack. And so we might predict, from what we can see in a static way before it's attacked, that this castle would not be very resilient. And we might be right, but there could be mechanisms that come into play and can be measured only when the castle is attacked that might give it resilience that we couldn't know when we just observed it in a static sense. So for example, if this castle has an amazingly trained troop of archers that can pop up into the windows and start shooting arrows, that might be like an incredible immune system that can come into play at the time of exposure to an infectious agent or a microbe and win the day. Likewise, if this castle has incredible stores inside of resources like food and lamp oil that could help it outlast a siege, that might be like having incredible nutritional stores or incredible abilities to be able to manage energy and utilization and metabolism. And a person who has that might be able to survive and get through and recover from a health stressor better than someone who didn't, even if they looked before like they had sort of the same level of structural frailty.
The same graphic taken from the Duke Pepper Center Conceptual Model of Physical Resilience, as shown in slide 11, depicting PreStress Reserve, Stressors, Resilience, and Outcomes within the environment.
So again, we sort of think of the pre-stress reserve as like the spectrum between how robust to frail the person is in this kind of static sense that could be measured before the stressor. We think of resilience as really being driven by these kind of dynamic processes that are important at the time of a stressor. And we've tried to set up studies to be able to examine and measure both of these things. So let me get into, first of all, how we try to quantify, did this person have a resilient response after the health stressor or did they not? So in thinking about this, we first thought about our two kind of key clinical questions when we're thinking about a person who, say, is facing an elective surgery or has just had a hip fracture. So first, we might ask ourselves, well, what's just the pattern of recovery that they would have? If I measured their mobility status or other sort of measure of health or function multiple times, what would the trajectory look like if I drew it? Another one is, we often think, how much better or worse do we think this person is going to do than expected, just based on what I kind of know about them, maybe their age and their chronic conditions and their functional status before they're stressed. So the first way,
graph depicting the recovery phenotype with time on the x-axis, baseline 2 months, 6 months, and 12 months, and variable score on the y-axis. An arrow indicates that stressors are introduced at baseline. The Journal of Gerontology, Series A, Biological Sciences and Medical Sciences, March 9, 2020, Volume 75, Issue 4, pages 731 to 738.
Is really just what we call the recovery phenotype approach. And essentially, we're trying to phenotype the recovery pattern. So we take some sort of a measure of health and we measure it before the stressor and then as many times after the stressor as we can. So, and on a time scale that makes sense for that measure of health. So for example, if the stressor was a hip fracture, we might be interested in somebody's mobility and we would draw it out. Likewise, we might be interested in something else like their cognition. So we would measure their cognition at multiple time points, or we might be interested in their depression or symptomatology and would measure that at multiple time points. This is very descriptive. We can do it with multiple different parameters and then try to summarize these multiple outcomes through something like doing a factor analysis or a principal components analysis after doing a latent class trajectory analysis, so that we can group people as a class that represents a great recovery versus a poor recovery. And the downside of doing it this way, the upside is that it's very practical and accessible, I think. The downside is that it is very driven by those pre-stressor variables, such as their age and their comorbidities. And this is one study where we did this in the secondary data analysis of people who had undergone a hip fracture.
Images of 8 graphs depicting the lowest, medium, and highest resilience data of the latent profile analysis trajectory group. Each x-axis measures months post-surgery. The y-axis categories include the following health measures: steps per day, Yale score, number of LE activities of daily living or ADL disabilities, grip strength in kilograms, seconds to complete 5 chair stands, gait speed in meters per second, number of instrumental activities of daily living or IADL disabilities, and balance with Z-score. Journal of American Geriatric Society, December 2019, Volume 67, Issue 12, pages 2,519 to 2,527.
And what you can see here is that we looked at a lot of different measures of health measured at multiple time points after the hip fracture. We looked at their number of step counts a day measured by actigraphy. We looked at things like gait speed, their self-reported function, their grip strength. And what was interesting about this was that no matter which of these kinds of measures we use, the latent class trajectory analysis sort of grouped people about the same. We had really good responders, medium, and not so good. The models found those same three groups no matter which measure of health we looked at. But
slide titled, What Factors Were Associated with the Phenotype of High Resilience After Hip Fracture? Featuring a bar graph measuring stressor factors, environment, psychosocial, comorbidities, demographics, and pre-stressor function. The x-axis represents model predicting high versus low/medium resilience, and the y-axis represents AUC for variable chunk. Stressor factors are 0.6, environment is 0.6, psychosocial is 0.61, comorbidities is 0.67, demographics is 0.67, and pre-stressor function is the highest at 0.84.
Then when we sort of put it all together into one summative measure, what we found, and we asked, what predicts whether people are going to be in that high resilience group versus the low or the medium resilience, by far the most informative thing was their pre-stressor function. And that was really perhaps not that surprising. So the next way that we tried to think about it was, well, okay, what if we tried to use all of those things that we know about their pre-stressor function?
Graph depicting the expected recovery differential with time on the x-axis, baseline, 2 months, 6 months, and 12 months, and variable score on the y-axis. An arrow indicates that the stressor is introduced at baseline. The predicted outcome, actual outcome, and expected recovery differential all occur at 12 months. The Journal of Gerontology, Series A, Biological Sciences and Medical Sciences, March 9, 2020, Volume 75, Issue 4, pages 731 to 738.
We predict for every person how they're going to recover based on those things that we knew about them at baseline. And then, so we essentially make a predictive model based off of a cohort of people who have all experienced this stressor. We, for each person, use our predictive model to calculate what we predict their outcome would be at 12 months. And then we look at what their outcome actually was at 12 months or another time point. And the differential between what their outcome actually was and what we predicted it was based on factors that we knew about them beforehand. And again, this is in the context of sort of one cohort or one group of people. That difference, what we hypothesized, is that might actually give us more information about those biological or sort of cellular, molecular mediated things that cause a person to respond much better or much worse than we would have expected just based on pre-stressor status. So we did a proof of concept to see if that was in fact the truth.
The word resilience is depicted in a central bubble with the following terms connected in a ring around the central bubble: response to damage and toxins, mobilizing energy stores, regulating inflammation and immune response, cellular damage control, protein folding and recycling, stem cells and regeneration, and genetics and DNA repair. Beside the circle labeled genetics and DNA repair, the marker for gene expression is the miRNA panel. Beside the circle labeled mobilizing energy stores, the markers for metabolism are acylcarnitine, branched chain amino acids, and IGF-1. Beside the circle for regulating inflammation and immune response, the markers for immune response are IL-6, IL-10, V-CAM, TNF, R1 and 2, and SASP panel.
We picked 3 of those pillars of aging, so biological pillars, biological processes that we hypothesized would be the ones that would especially come into play when the castle is attacked. So the ability to mobilize energy stores, the ability to regulate inflammation and have an adaptive immune response, and the ability to repair your DNA or to activate and necessary genes at the time of a stressor. We looked at markers of each of these sort of biological pillars of aging, and we asked the question, does it predict resilience as defined by this expected recovery differential?
Slide titled full biomarker panel explains 38% of resilience variance in a hip fracture cohort, depicting a line graph, a bar graph, and the final LASSO regression model in a table. The Journals of Gerontology, Series A, Biological Sciences and Medical Sciences, September 25th, 2020, Volume 75, Issue 10, pages E166 to E172.
And in a fracture cohort, we actually found that our biomarker panel explained 38% of the resilience variance. Now, to the people who do biomarker studies, this seemed like a huge amount. They were so excited. They're not used to a biomarker, an assay-based biomarker, predicting that much of a clinical outcome. Even in this kind of parsimonious, reduced set of biomarkers that we got from lasso regression, it still explained 27% of the variance. So in some ways, we felt that this did, in fact, fit with our hypothesis and, in fact, generated new hypotheses about which biomarkers seem to be the most informative.
The slide shows a graph depicting the level of function on the y-axis and the recovery after fracture on the x-axis, and shows the biomarkers associated with physical resilience, miRNAs, free amino acids, acylcarnitines, IL-6, TNFR-1 and 2, SVCAM-1, IGF-1. Low resilience is associated with increased cellular senescence, increased inflammation, decreased mitochondrial function, and decreased skeletal muscle metabolism. Daniel Parker, MD.
And at the end of the day, it seemed like the ones that were the most predictive of high resilience, which here I'm defining as the person recovering better than you would have predicted. Those people tended to have lower markers of cellular senescence, lower markers of chronic inflammation overall, higher markers of better mitochondrial function, and higher markers that suggested that they had better skeletal muscle metabolism. So again, it sort of fit with what we would have predicted. So that's a lot of how we've been trying to sort of measure, were people resilient or not. Then we've also turned attention to how we might be able to predict resilience, and we focused a lot on provocative tests. So what do I mean by provocative tests? What I mean is stress tests, essentially. So where you measure the response to an experimental stressor and you see if this might actually predict their capacity for resilience for real world stressors in the future. So some of the common ones that are used all the time in clinical practice are like a cardiac stress test, the glucose tolerance test, where you give somebody a measured amount of glucose and it predicts how they would respond to, a real world meal, and if they have diabetes, and then dual task tests. So what do I mean by a dual task test? So I love these tests.
The slide depicts a bar graph measuring gait speed in meters per second between low-risk and high-risk groups, and a box and whiskers plot measuring the dual task effect on gait speed between low-risk and high-risk groups. Whitson et al., Dual Task Gait and Alzheimer's Disease Genetic Risk in Cognitively Normal Adults, a pilot study. Journal of Alzheimer's Disease, 2018, Volume 64, Issue 4, pages 1,137 to 1,148.
They kind of are based on this premise that if all of us, when we multitask, we tend to sort of screw up both jobs. And in this case, we give people two very easy tasks to do. We look at their performance when they're just doing the task by itself, and then we make them do those two things at the same time. So it's kind of like a test that might be asking, can you walk and chew gum at the same time? In this case, we're having them walk, and then we have them do a cognitive test. And the cognitive test is often based on response time to getting questions answered accurately. When we make people do these two things together, we expect that most people do a little bit worse on each task, but the people who do a lot worse, doing a lot worse, tends to predict worse outcomes. So what I'm showing you here is data from a pilot study that I did with dual tasking, where I took people who did not have Alzheimer's disease, were cognitively normal, had no subjective symptoms, but these were people in their 60s, And they had agreed to be genetically tested for Alzheimer's disease risk genes and to not know the results. So some of them, we picked a cohort that had 14 people that carried an ApoE4 gene, or the highest genetic risk factor for Alzheimer's disease. Then we had 14 age-matched controls that seemed exactly the same as those other people, but didn't have an ApoE4 gene. And what we saw was that when in the single tasks, both groups performed the same way. But when we put them in the stress condition of the dual task, that's where we saw a difference between the groups. And the people who are ApoE4 carriers, or the high risk group, showed greater decline in their walking speed when they were forced to walk and do cognitive tests at the same time. So the idea here is that by increasing the sort of demand on their brain, you are evoking a phenotype that potentially suggests their risk of Alzheimer's, or even early Alzheimer's, that they could be harboring, that is preclinical. We also do sometimes ex vivo provocative tests. The idea here is that with a provocative test in a very frail older person, there's a little bit of danger in just doing the test. You could potentially be trying to provoke them in an experimental way and actually do them harm. So we don't like to actually expose people to things like toxins. But what we do in this case is we expose their blood instead. So we draw the blood into a tube that contains the toxin. So we've done it with, it can contain a flu virus or a flu vaccine, or it can contain the LPS toxin. And then we look at how the blood, the white blood cells, or the peripheral blood mononuclear cells, how they react to that toxin. And we look at whether those that mount a greater response potentially predict how these people will respond to real world stressors. And it's a way to stress the person without putting them in any danger.
Peripheral blood mononuclear cell reactivity is quantified as the difference in cytokine and granzyme B, or GRB, activity from stimulated to non-stimulated conditions. Cytokine level of IFN gamma, TNF alpha, IL-1 beta, IL-6, IL-10, IL-15, and IL-22 in the supernatants of cell cultures from peripheral blood.
And then these are a couple of examples of studies where we're trying to sort of predict physical resilience after health stressors. So the first one that I'll tell you about, and I'll tell you also, I'm expecting that we're going to have about 15 minutes at the end, and I would love to get your questions and thoughts. So the first study is a pilot study that we called the PRIME EEG study. The idea here was that we did a 32-channel EEG on 50 individuals that were 60 years or older and were facing an elective surgery. And we took all different kinds of surgery, except none of them could be neurosurgeries or cardiac surgeries. So most of them were abdominal surgeries. Some of them were thyroid or parathyroid surgeries. And we also took orthopedic surgeries. So before the surgery, we took them in a resting state with eyes closed and then eyes open, and we got an EEG of their brain activity. And then throughout the surgery, they also wore the EEG cap, and we selected 3 minutes per protocol, and then we compared the EEG patterns when they were at rest before the surgery, and then during the surgery, which in this case, we were using the surgery almost as our experimental stressor to sort of then see if we could predict from what their brain waves did during that stressor, how they would recover in the days after surgery. And then we analyzed the data from the frontal leads, and we were interested in sort of how the complexity of the EEG pattern changed. And we measured the complexity by this thing called multiscale entropy, where you essentially calculate something called entropy, which gives you a sense of the randomness within the data. And we calculated it at different time scales, meaning first we used the very coarsest time scale that we could get, which was 200 milliseconds. So we sampled the data every 200 milliseconds and calculated the entropy based on that sampling. Then we did a sample that took every other measurement. So rather than every 200 milliseconds, you're looking every 400. And you go at these increasingly sort of coarse timescales.
Acker L et al. Electroencephalogram-based complexity measures as predictors of postoperative neurocognitive dysfunction. Frontiers in Systems Neuroscience, November 10, 2021, Volume 15, pages 718 to 769.
And what you can do is plot these sort of plots that plot at the different scales that we calculated the entropy.
Slide depicting two graphs. The first is a box and whiskers plot measuring EEG complexity with preoperative versus intraoperative represented on the x-axis and area under the MSE curve represented on the y-axis. The second is a line graph measuring EEG complexity with MSE represented on the x-axis and mean frontal complexity represented on the y-axis. Preoperative versus intraoperative are differentiated by color. Frontiers in Systems Neuroscience, November 10, 2021, Volume 15, pages 718 to 769.
What was the entropy? The interesting thing that we found was that overall the complexity got higher during the surgery, during the time that the brain was being exposed to anesthesia. But what we observed was this very unexpected thing of what we called the crossover point, which means that the preoperative complexity was actually higher when we calculated the entropy at the shortest timescales, but the opposite was true when we calculated it at longer timescales. And most of the time, the complexity was higher during the surgery. So this single clear crossover point was present in 42 out of 50 people. In other people, the lines crossed multiple times, or in a few cases didn't cross at all. And what we thought was interesting about the crossover point was that it seemed to be normally distributed and consistent within a person. So within a person, it didn't matter where we sampled the EEG data from within the surgery. We always sort of got that person got the same crossover point calculated. But it was a normally distributed thing amongst the people who had it, which made us think that it may be something biologically meaningful. And in fact, what we found was that with borderline significance in this small pilot study, it did in fact predict their change in delirium scores and change in attention scores on the day after. So the hypothesis that sort of came out of this was that perhaps the location of where that crossover happens may be a reflection of the integrity of sort of neural control mechanisms in the brain. It essentially, the scale at which you see equal complexity intraoperatively and before may correlate to how much integrity the brain had and how well it withstood the stressor of anesthesia, basically. So there's a lot of ongoing work. This is the team here. It's led by a team both at Duke, and then our collaborator, Oolohipsis, is at Harvard. The question is, are there other EEG-derived metrics besides entropy that may predict delirium? Other groups around the country are asking that same question. We're really trying to understand, what does that crossover phenomenon mean? What does it represent physiologically? And then is there a way to collect perioperative EEG data that may be clinically informative, but is easier, can be more feasibly cleaned and simplified? So finally, I'll tell you too about the Prime Knee study, which is an ongoing study. We don't have results yet, but it's enrolling 250 people who are scheduled for elective knee replacement surgery. And we collect a lot of data on these people. We are currently at about 110 people enrolled. We've had a little disruption due to the Omicron variant, but we're back up and running now. But at baseline, we collect information about folks' cognitive reserve, physical reserve, psychosocial reserve, and then also we do a series of provocative tests. We collect blood for biomarkers, and then we monitor people's step counts throughout. And then we collect over time at these monthly phone calls and quite a bit of data right around the week of surgery, because what we want to do is to be able to sort of draw those recovery phenotypes and trajectories. And then one of our main questions is which biomarkers and which provocative tests are best going to indicate the resilience after the surgery.
Whitson et al. A template for physical resilience research in older adults, methods of the PRIME-NI study, Journal of the American Geriatric Society, November 2021, Volume 69, Issue 11, pages 3,232 to 3,241. After the baseline visit is surgery week, during which a provocative test is done, ECG variability. Covariates include surgery characteristics, complications, and the length of stay. Pain intensity and interference are measured daily. 3D CAM attention items, blood for biomarkers, and 7-day step counts are all collected and recorded. After surgery week are phone calls at 1, 2, and 4 months after surgery. Measures include pain intensity and interference, LE, PADLs, or LE paddles, cognitive change index, subject informant, 7-day step counts, and intercurrent events and rehabilitation received. The process concludes with a 6-month visit during which cognitive and physical reserve tests and provocative tests, including dual task gait/cognition, functional near-infrared spectroscopy, or FNIRS, reactivity tests, and the PBMC reactivity tests are performed. A final 7-day step count is also recorded.
These are some of the measures that we use to try to assess their pre-stress reserve. So we have, again, physical reserve, cognitive reserve, and psychosocial reserve that we try to sort of measure statically before they're stressed. I'll note that one of the ones that we use for psychosocial reserve is the resilience scale. It's a 25-item scale that sort of captures the construct of resilience that has been well developed for a long time in psychological or psychosocial literature about how we sort of cope at a psychological level to stressful events. And we think that that's incredibly important to how people will respond physically to a health stressor.
Also included under the psychosocial reserve is the PHQ-9, which involves participants indicating how frequently they have experienced 9 depression symptoms over the last 2 weeks to measure depression associated with function in multiple domains. Emotional support is also included, which involves the PROMISE emotional support short from 4A for participants to report how frequently they have access to social support when they need it. Never, rarely, sometimes, usually, or always. This measures the strength of patients' social support networks. The physical reserve includes a 3-minute walk during which participants cover as much distance as safely as possible over 3 minutes. This is the shortened version of the standard 6 minutes for the severe knee arthritis population. This test measures aerobic and musculoskeletal reserve. Also included is grip strength, which involves a single trial, dominant hand via a handheld dynamometer using a validated protocol. This test measures muscle strength. The cognitive reserve includes 3MS, which is a screening instrument with 30 items, with a score range from 0 to 100, assessing cognition in 5 domains: orientation, registration, attention and calculation, recall, and language. This measures global cognitive impairment. Trials making test A and B are timed tests for attention and concentration, requiring subjects to draw lines connecting circles containing numbers or alternating numbers and letters in sequential order. This measures complex executive functioning skills such as visual attention and logical task switching. The 15-time word list is Form 1 of the Ray Auditory Verbal Learning Test, 15 words, list verbal recall, scored as % of words correctly recalled. This measures verbal list learning and memory recall. Finally, the digit symbol substitution test involves subjects drawing corresponding symbols below a set of numbers in the order listed by referring to a key at the top of the page. This test is timed and is conducted to measure sustained attention, visual spatial skills, and set shifting.
Then we do the provocative tests. So we do a dual task test. We look at their cerebrovascular reactivity in response to a few stressors. That's what you see here. I have this person hooked up to an FNIRS, or a functional near infrared spectroscopy, which is kind of a poor man's fMRI to look at what blood flow is doing in the frontal lobes. We do EKG and heart rate variability, and then we do those white blood cell tests where we stress the white blood cells after they're already out of the person's body. I'm going to conclude with this is a participant because I always like to bring it back to the patients. This was a gentleman who finished our study and was obviously sort of a poster child for good resilience because we actually had a hard time getting in touch with him to schedule his 6-month visit because he was always on the golf course. So I had to ask him about.
So as you know, one of the things in this study that we're trying to understand is why some people do really well after surgery, like you did. Some people don't do as well and bounce back as well. I wonder what your thoughts are about what do you think are some of the things that help a person be resilient after they have a surgery or an infection or any other kind of stressor?
Well, my situation was, since it was a knee replacement, I felt that my biggest thing was just pain endurance, endurance that pain. And once within the first week, myself, I like to consider myself, I'm gonna always go to the extreme when I'm trying to do something like rehab and physical activities. So within the first week, I was up walking. At first I had a little walker, and then next couple of days I threw the walker away and just limping around. And
so again, I think what I hear in what he's telling us is that he's placing a lot of importance on his own personal grit and motivation, and desire to get better. And I think that obviously, as much as as a sort of biologist, I like to sort of think about the molecular, the cellular mechanisms that probably also helped him get better. I think he's absolutely right that his own personal grit and fortitude had a lot to do with it. At another point in the interview, he also credited his wife, sort of saying that she was a little bit of a taskmaster, sort of a drill sergeant about his physical therapy. So I think, again, the importance of social support can't be underestimated.
Slide featuring an image of Vasily Kandinsky's painting Rotenflock auf Raumen and a painting by Urs Verli that displays all of the shapes of Kandinsky's painting sorted by color and size.
And I'll just end with this. I always feel like that there's something about resilience or recovery from a stressor that feels so poetic and so it's quintessential to the human story and the human narrative. And yet I feel like that by trying to study it, we necessarily have to kind of break it down. I was introduced to this artist by Steve Krzyzewski at Wake Forest, who likes to show this too. And Urs Verli is a sort of comedian and artist who says that he tidies up great works of art. And here's his take on Kandinsky. So I am very aware, I think, as a scientist, of how it sometimes feels like we're deconstructing something that should maybe not be should be appreciated in its full and human form. But I also think that there's some necessity and also some beauty in trying to come up with these constructs and concepts to be able to advance the field. I just want to again thank the collaborators who make this resilience work possible. There are many at Duke, and also our collaborators at University of Maryland, Connecticut and Jack's Labs, our Harvard collaborators, and close collaborators at Johns Hopkins who similarly focus a lot on both frailty and resilience. And then our collaborators at the NIA, Giovanna Zappola, Basil Eldada, and Chanda Dutta, who have really stimulated this field and helped to elaborate and been really strong partners in how to think about and apply resilience.
Duke collaborators include Kathleen Colin-Emmerich, Ken Schmader, Kim Huffman, Bill Cross, Virginia Cross, James Bain, Micah McLean, Miles Berger, Marty Waldorf, Daniel Parker, Janet Hubner, Harvey Cohen, Miriam Morey, Carl Piper, Rick Sloan, Mary Cooter, Jody Feld, Patrick Smith, Catherine Hall, and Leah Acker. University of Maryland collaborators include Jay Magaziner, Denise Orwig, and Ann Gruber-Baldini. University of Connecticut and JAX Labs collaborators include George Kuchel, Jacques Banchereau, Janet Michelini, and Doigu Uchar. Harvard collaborators include Lou Lipsitz and Zhenghong Zhao. Johns Hopkins collaborators include Jeremy Walston, Ravi Varadhan, Karen Bandine Roche, and Peter Abadir. NIA collaborators include Giovanna Zeppola, Basil Eldada, and Chanda Dutta.
So I will go ahead and stop. That little creature there is a tardigrade. It's supposed to be the most resilient creature known on the planet. It's a microscopic creature that you can freeze it, starve it, dehydrate it, and it will bounce back. So that's our mascot.
Thank you, Dr. Whitson. What a wonderful presentation, so full of so much information. I really appreciate you joining us today. I'm going to start with one question, and I haven't been checking the chat box because I can't walk and chew gum at the same time. But so I don't know what that says about my resilience, but I do have one question, but then I will call Dr. Barbara Cohen to help us to sort through the questions in the chat box and guide us through from there. My one question, since the resilience program at NIH is led by the Office of Dietary Supplements, we can't go without asking the question of how might an intervention like a dietary supplement either enhance resilience or go back to that reserve. So you talked a lot about the reserve point being the predictor as to whether or not you would have a resilient outcome. Is there any possibility or any of your studies that have shown that an intervention such as a dietary supplement might be able to boost that reserve in some way?
Yeah, so I'll note here two of the dietitians who do a lot of this research and are very involved in the program. Connie Bales and Katie Starr are two dietitians that ask this question a lot, and I don't know that we found what I would say like the magic bullet, but certainly a lot of their work has suggested that in general, for example, if you take particular stressors like heart failure, they've looked at heart failure populations, and a lot of the response to a new diagnosis of heart failure in terms of how it affects people functionally can be predicted by that sort of nutritional reserve measured in a few different ways. And so I think that is an incredibly hot area, and a lot of those pillars of aging, the biological processes, very directly relate to diet and nutrition opportunities. So certainly metabolism is one of them, and energy utilization. But then also when we think about things like oxidative stress and how we might reduce oxidative stress, that would have a favorable effect in at least one of those pillars, if not others, in terms of DNA damage and other things. So I think that's an absolutely critical area for the future.
Great, thank you. Barbara, would you mind? Sorting through the chat box.
Green, but yes. So I think just sticking with the idea of an intervention, somebody asked, did you have a chance to try whether you can improve the postoperative EEG response with some intervention pre or peri during the procedure, such as music, meditation, or something along those lines?
That's a really interesting question. We did not in that particular study do any kind of intervention. It was that pilot study was just observational. One of the things that we did try to look at was whether the choice of anesthetic agent might have, play a different role. And among the anesthetic agents that we included in the study, it wasn't obvious in that small pilot study that one was associated with a better crossover point than others. But I think that that's a question that's really worth considering too. And that question about, we do have someone that studies not music exposure during during a surgery, but in people who are in the hospital, either from a medical illness or from a surgery, she's testing a music intervention particularly to try to prevent delirium.
Great, thank you. Could you explain more about how you calculate the latent class trajectory and the lasso regression?
So the answer would be, I wish I could explain more. I'm probably not the best person to try to explain. We have a data science and statistics lab, and they could spend an entire hour explaining. So I have a rudimentary understanding of the statistics that support it. The idea, though, is for the latent class trajectory analysis, the idea is that each person is sort of assigned a trajectory or a slope, and the slope doesn't have to be, the terms could be added to describe the if it's not a linear trajectory. But each person gets sort of assigned a trajectory, and then the latent class part of it is essentially using an empiric approach to try to sort people into different class matches. So grouping different trajectories together. Lasso, as my understanding is, it really is just used to find the most informative of the independent variables or the covariables that are predicting some outcome.
Okay, great.
You have to have my statisticians back if you want much more detail about that.
We'll go away from statistics. And how do non-individual factors contributing to resilience, such as social support, environmental factors, fit into your model of resilience?
Yeah, so that's a great question, and an area that we are actively trying to expand, because to be honest, we're mostly very clinical, translational, biology focused people, and so we see this as an area that we need more expertise and partnership. What we have done is tried to measure using sort of off-the-shelf existing indices to measure different constructs, but we are very open to collaboration to help us capture that piece in a more informative way, because or just new ways, because I think that it is such a critically important piece.
Okay, great. Are there CNS or other physiological correlates of positive effect or grit, so to speak?
Yeah, so grit is something that I'm really interested in. That would be one of those tools that we've used, but I don't know. That is one of the questions that we're going to be asking, is what are some in the PRIME study, for example. What are some of the physiological measures that we're collecting, both the dual task performances and the other provocative test performances, but also the biomarkers? Which ones seem to relate most strongly to these kind of psychological measures? I think is an open and really interesting question. And also how those psychological measures change, because that's something that we're looking at too, is the pre-stressor status, but we also collect that data again 6 months after.
Great, thank you. What type of cognitive task can be used in conjunction with walking to evaluate decreased speed?
Yeah, great question. So there's whole review articles on which cognitive task to choose to go into your dual task. And the one that we have found that is has the largest effect on the gait speed, so we like to use it, and it's also very portable and feasible for clinics, is simply sort of speech production. So what we do is we cue people to tell a story, and we ask them to talk for about a minute, and we give them cues like, for example, tell us about your family's favorite holiday and what your traditions are. And then they walk, and while they walk, they talk and tell us that story. Or the cue might be, tell us about a vacation that you took, or a vacation that you'd like to take, and what you'd like to do on the vacation. So we give them something where they could talk for about a minute, and then we just let them go. And we use that one a lot because it has one of the largest dual task effects on the gait speed. One of the challenges of it is it's harder to quantify performance on that cognitive task. So another one that we will often use is, for example, and I wish I had the little video in here that shows somebody doing it, but it's sort of like if you're familiar with a Stroop test, it's kind of like an auditory version of it. So when we do this, we have a person and they're usually walking and they have a headset on. So we're recording what they say in the headset, and then we can grade the cognitive test later. But they get these cues, and first, the easy version is you ask people, when I say green, you say go. When I say red, you say stop. And then you switch it on them and you say, now when I say green, I want you to say stop. And when I say red, you say go. So it's kind of the opposite of what we're trained to think. And it gets it kind of executive function and processing speed. And that test can be graded both for accuracy and speed of response when we look at the recordings. There's lots of others. You can have people count backwards by threes from 40. There's lots of other tests that have been used.
How is ADD, or attention deficit disorder, accounted for in your dual task stressor measure? Would one with ADD be slower?
Yeah, so that's a great question. We don't account for it directly, but in a sense, every person serves as their own control, because we're always comparing how they performed on that task as a single task to how they performed on that task having to do both things at the same time. So it is possible that we'd see a greater dual task effect in people who have ADD. One might hypothesize, and there may be somebody out there that has data to say exactly that. I actually don't know if dual tasking has been tested in ADD patients or as an indicator of ADD. I usually think of that as something that pediatricians or people that study the earlier end of the lifespan usually study more, although certainly older people with ADD get old. So I have patients with ADHD too.
True. Do you ever use the 6-minute walk test, which is widely used in cardiovascular research?
Yeah, so in the PRIME study, we modified it to a 3-minute walk test, partly because for the people are about to have a knee replacement. So a lot of them get into a lot of pain, even sometimes by the end of 3 minutes, and 6 minutes in our pilot study just proved to be too much for most of them to do. So we modified it to a 3-minute test. But in other protocols that are going on in the Aging Center, we use the 6-minute task a lot.
Okay. Have you found that resilience can be developed, or have you assumed that it is mostly genetic and based on healthy lifestyle behaviors? Can wellness coaching make a difference?
Yeah, what a good question. And I think in some ways it depends on what kind of resilience we're talking about, and maybe that's something I think is great about this group and having this special interest group is, I think there are probably some aspects of resilience that are very coachable, and that we can improve people's resilience. There are probably some aspects of resilience, those things that are maybe more molecular or biological, that some of those might be genetic and we're born with them, or it's luck of the draw. Others might be modifiable, but maybe not through coaching. But I certainly think that a lot of resilience is modifiable, and honestly, I think it's the reason that I'm so interested in it and want to study it. I think we could prepare people much better. There's a see how much time we have. I know we're at the end of time, but there's a metaphor that we often use with patients that I borrowed from a guy named Craig Weller, who studies resilience and is a physical therapist, but talks about that he used to tell people when he was training them in a military context, he would almost like scare them on the first day by telling them that they were going to have to go out in the ocean with nothing but a raft, and he didn't know what could come. It might be sharks, it might be bad weather, it might be any number of things, but the only thing that they could control was the raft, and the military would spare no expense in helping them build a raft to their specifications and any material that they wanted, and they should think about how easily repairable it was and how sturdy it was. And then he would tell them, okay, relax, you're not actually going to have to do that, but the raft is your body and mind, and you are going to go out and face things, and I can't tell you what kind of stressors might come your way, but what you can do is ask yourself every night, what have I done to prepare my raft, meaning my mind and body. And so to that extent, to the degree that I think I've seen patients find that also very motivating to think it's true. You know, as I get older, stressors are going to happen, and I'm going to get diagnoses and have events, and the best thing I can do is just try to make my raft as sturdy and easily repairable as possible.
Thank you, Dr. Whitson. That's a wonderful way to close a wonderful seminar presentation. I saw lots of thank yous and applause in the chat, and so I just wanted to pass that along to you. We're going to close this seminar series with an announcement that the next seminar, I believe, is scheduled for, oops, sorry, I should have had this ready. It's scheduled for Wednesday, May 18, 2020, and the presentation title will be Slowing Down Fibrosis in the Aging Heart. The speaker will be Dr. Mark Entman. And if you want to give your applause by way of your chat or the little emoji to Dr. Whitson, that'd be great. And for now, we're going to close out the Trans-NIH Resilience Working Group seminar series. Thank you again, Dr. Whitson.
Thank you. Thank you so much.